the nervous system. Neuroglia, literally translated, means nerve. These again are the supporting cells of the nervous system. The supporting cells are indicated here. There are astrocytes, these star-shaped cells. Oligodendroglia or oligodendrocytes, these are cells that myelinate the axons of neurons within the central nervous system. The Schwann cells myelinate the axons of neurons within the peripheral nervous system. Microglia are phagocytic cells, and they can either be stationary within the central nervous system. They can be roving, for example, moving around and collecting cell debris. And then ependymal cells, which by definition aren't true neuroglial cells because they don't have any direct supporting function of a neuron. These are cells that line, for example, the central canal that runs down the middle of the spinal cord, that will line ventricles within the brain. They're columnar epithelial-like cells that have cilia on them, and their primary job is to circulate cerebrospinal fluid. The main type of neuroglial cell is an astrocyte. So around 75% or so of neuroglial cells will be astrocytes. And we'll talk about some of these in the context, for example, of cancers, which brain cancers are uncontrolled growth of neuroglial cells rather than neurons, and most of those are referred to as astrocytomas. We also have ependymomas, for example, the overgrowth of those cells that line, say, a ventricle. We'll talk about microglia and the pathophysiology of diseases like amyotrophic lateral sclerosis, for example, which they can actually start to destroy cells that are healthy, and in the case of amyotrophic lateral sclerosis, motor neurons, etc. These points of contact between neurons are known as synapses. A synapse comes from a Greek word literally meaning to bind or to join. Neurons are bound or joined to other neurons or to muscle cells by these particular connections. However, there's a space between neurons. They're not physically connected together, nor are they physically connected to a muscle, and that space is known as the synaptic cleft. At the end of a neuron, the axon actually widens a bit. You'll see this in the next figure. The reason it widens a bit is because there are a high number of mitochondria at the end of the axon, suggesting a high amount of energy that's required at that particular point. And the ends of axons also have a very high quantity of synaptic vesicles. These synaptic vesicles contain neurotransmitters. There are dozens of identified neurotransmitters. We've talked about a number of these already, from dopamine to GABA to serotonin to norepinephrine. These neurotransmitters can be considered excitatory or inhibitory. When a neuron releases a neurotransmitter onto another cell, what it's going to do is either increase or decrease the likelihood that that other cell is going to produce an action potential itself. If the neurotransmitter causes what's known as an excitatory postsynaptic potential, that's something that's going to move the membrane potential of that particular neuron from its resting level to something that's closer to threshold, something that increases the likelihood that that neuron might produce its own action potential. If a neurotransmitter is inhibitory, that is something that would cause, for example, the membrane potential to become more negative, something that moves it away from the threshold level, thereby decreasing the chance that the neuron is going to undergo an action potential. Because of this convergence and divergence that happens in the nervous system, we don't have this one-to-one connection between neurons as we do, say, in a reflex arc, for example. Many neurons are dependent on something known as summation. There's something known as temporal summation and spatial summation. Temporal refers to time. So if one particular neuron continues to produce an action potential and release neurotransmitter onto another neuron, it's something that could move that potential of that neuron to a point that's closer to threshold and result in the production of an action potential. Spatial summation refers to a number of different neurons converging onto one neuron. all producing a change in membrane potential that might then lead to the reaching of that threshold value, therefore the production of an action potential. So these synapses are dependent on summation. You can have temporal summation, spatial summation, you can have summation of inhibitory signals, you can have summation of excitatory signals, you can have both of these signals occurring at some particular time, resulting in a balance, etc. A little more detail of a particular synapse, and here's a certain type of synapse that between a motor neuron and a skeletal muscle. This is known as a neuromuscular junction. Remember that motor neurons, and here we're talking about neurons in the somatic nervous system, are going to release the neurotransmitter acetylcholine. So what we see at the end of this axon, you can see the axon is covered by a Schwann cell, which is going to produce that myelin sheath, that insulating layer around the axon. You can see that this axon is wider at the end than it is in the middle. It contains these synaptic vesicles, which, along with the mitochondria, lead to the end of this particular axon being wider than the rest of it. These synaptic ends are also referred to as synaptic boutons, and that was a term that was mentioned in the prior slide, or indicated in the prior slide. Literally means button. It has this button sort of shape if one were to look at it from the underneath, and you can see that via certain types of microscopy. So again, you can see it's wider than the rest of it because it contains these synaptic vesicles and mitochondria. Here we see this particular motor neuron releasing acetylcholine onto this skeletal muscle, that neurotransmitter diffusing across the synaptic cleft and binding to acetylcholine receptor sites. Acetylcholine is going to cause then, if there's enough of it, this particular skeletal muscle to contract. And there are some pathophysiology that we'll talk about in the context of this, like in disease, myasthenia gravis, for example, what can happen in a number of different types of paralysis in which you have failures of this acetylcholine system. Next up, the meninges, this protective layer that's around the brain and spinal cord. So here we see a coronal section through the head, and you can see brain at a particular level, indicating the third ventricle and the lateral ventricles, and the corpus callosum, this bundle of axons that is connecting the two cerebral hemispheres together. And then around that is going to be a space that will be filled with fluid, cerebrospinal fluid. This clear, colorless fluid, very similar in composition to blood plasma, and that's indeed where it comes from. And then we can see layers that are a little bit more about cerebrospinal fluid. Again, clear, colorless fluid, very similar in composition to blood plasma, although it's a little more viscous than blood plasma. There's not all that much of it, but it's constantly circulating within and around the brain and spinal cord. It's produced within the lateral third and fourth ventricles of the brain by structures, these tufts of blood vessels known as choroid plexus. See one of those when we talk about, for example, hydrocephalus, in which there can be a failure of movement of this fluid from choroid plexus into the portions of the ventricles as indicated. This fluid will then be reabsorbed through what are known as arachnoid villi, and again you'll see what can happen, pathophysiological consequences of this if there's blocking of those particular structures. Blood supply to the brain generally about 800 milliliters to 1 liter per minute. Primary regulator for blood flow to the brain is carbon dioxide, and that carbon dioxide will be detected by structures within the medulla oblongata, as we talked about before. In regard to the blood that does make its way to the brain, there is this barrier between blood and nervous tissue known as the blood-brain barrier that's actually made possible by a certain type of glial cell known as an astrocyte. Astrocytes literally hold capillaries next to neurons, and because they do this, they can actually regulate junctions between capillary cells. They can regulate what moves from within the blood into a neuron, therefore this blood-brain barrier. Primary blood supply to the brain via the internal carotid and vertebral arteries, and these will converge to form at the base of the brain something that's known as the circle of Willis, something identified by a British physician named Thomas Willis back in the 1800s. So here we can see the circle of Willis that's at the base of the brain. You can see the very prominent basilar artery that's indicated there on the bottom leading to the circle. We can see the internal carotids as you move in the anterior fashion, and then the circle that consists of the posterior communicating artery and the anterior communicating artery. A number of different branches off it that will then feed the rest of the brain tissue. You will not have to know these specific ones, but I might give you particular examples like this when, for example, there's occlusion of the anterior cerebral artery. There can be a loss of function in the area that's shaded in that light pink. You're seeing a ventral view of the brain indicated there. Occlusion of the middle cerebral artery is going to cause again cessation of blood flow to areas of the temporal lobe, for example. Occlusion of the posterior cerebral artery is indicated there. Occlusion of middle cerebral artery on that figure there on the lateral view, shown just like it is on that ventral view figure below, as well as the occlusion of the posterior cerebral artery, which is going to result in a failure of function of many parts of the occipital lobe and the temporal lobe. Blood supply to the spinal cord, made possible by the vertebral artery and the anterior posterior spinal arteries, as well as the aorta. Here we see that very prominent basilar artery again, and the branches that are coming from it, the anterior spinal artery, and there's the vertebral artery, that is the branch ultimately leading to that. As we look at the section through the spinal cord indicated there on the right, on the top of that figure would be the spinous process associated with the vertebrae. On the bottom would be the body of the vertebrae, and there we can see the spinal cord resting within it, and how the spinal cord will ultimately be fed primarily by the posterior spinal artery located near that spinous process, and the anterior spinal artery located near the body of the vertebrae. Here we can see some branches associated with it: posterior radicular artery, anterior radicular artery, interior spinal arteries, which will then ultimately feed the tissue of the spinal cord itself. There are, of course, a number of changes that happen in association with aging in the nervous system. A couple of these we've talked about before in regard to, say, overall brain volume, for example, which will drop normally from 10 even to 15 or so % with age. Brain being roughly about 3.5 pounds, 1,450 grams or so, that will steadily decline over the course of age, which, as we talked about before, unfortunately starts at around age 27 or so. Here we're talking about aging in the nervous system associated with those in their late 50s to 60s and 70s and 80s or so. Structural changes, like, for example, a decrease in the number of neurons, a decreased brain weight in size, as mentioned, increased adherence of the dura mater to the skull, which can then affect the underlying brain tissue. Thickening of the meninges, and the fibrosis and thickening of the meninges can actually be due to the loss of the cerebral tissue itself. Loss of cerebral tissue is going to be pronounced in regard to very narrow gyri, like sulci, so very narrow bumps associated with the brain, and grooves associated with it, with a corresponding increase in the size of the subarachnoid space, as one would expect if you have that change in those particular structures. In the basal ganglia and the ventricular system, there's going to be enlargement of the ventricles. There's going to be a decrease in size of these structures within the forebrain. A control motor response is known as the basal ganglia, and therefore you can see either due to or as a result of this change in ventricular system separations and vascular structures associated with the brain.